students. It's with great pleasure that I welcome you to the Department of Microbiology and Immunology. COVID-19 has already introduced you to microbiology in a big way. We already know that despite being at the peak of advancements, human beings can still be at the mercy of microbes. You know, you've been exposed to the most common way, most effective way of spreading of microbes, that's through air, through inhalation. And you've also been introduced to how to prevent so many infections through the various preventive measures that you have been practicing for so many days. What will we be covering in this lecture? We will be understanding what is bacteriology, a brief introduction. We will understand something called the Koch's postulates, concepts of vaccination and antisepsis, and of course, we'll conclude by understanding the morphology, biochemical characteristics, and genetic characteristics of bacteria. So, we need to understand where we've come from. We've come from a time when we did not know that there were microbes. And even when the microscope had been invented and people had seen tiny little animalcules, the correlation hadn't happened that these tiny little animalcules can actually cause disease. So, as late as the 19th century, you find this painting by Robert Seymour, and he says that cholera is caused by bad air, and that word is really miasma. That is, bad air leads to inhalation, or breathing in bad air is what causes all sorts of infections. And this is 19th century. Although, if you know, people knew about, people had been making hypotheses about all this ages back, but there was no proof. And even with proof, man had not correlated the facts. So it was considered that there is miasma and that is what causes disease. And then in the late 19th century, a huge debate came about. And that huge debate was, is the theory of miasma correct, or is there any weight or any truth in the theory of contagion which was coming up? So, till the late 19th century, miasma was considered dangerous in most Western cultures. And it was thought, as I told you, that disease is present in the air and proliferates without physical contact. And strangely, Florence Nightingale was a prominent member of this theory. And then we had contagionists who were more scientific in their approach, and they said, no, no, it's not possible that just breathing in air can cause disease, and there has to be some scientific basis for it. So it could be direct contact, it could be formalin, it could also be air. For example, now you know that COVID is spread primarily by breathing in infectious air, that is, somebody sneezes or coughs and he releases those microbes and the other person breathes it. So it can spread through air, but there are other routes like inhalation, ingestion, by inoculation, by contact, by fomite. But throughout the 19th century, the medical community was divided on the explanation for disease proliferation. Now we know that we have more germs than us. So the advances which have gone on, and with especially the genetic advances and the understanding of microbiota, what we now know is that of course we're a human being, but one, we are floating in a sea of microbes which we cannot see. You know that microbiology is a science of tiny little living organisms which we cannot see. Not only are we swimming in an ocean of microbes, we are also carriers of microbes. And those carriers are 10 times the number than the number of cells we have. So that means we are more germs than us. And when I'm saying hello to you, I'm actually saying hello to a lot of germs as well. So if we look at the origin of life, we realize that bacteria were the first life on earth. So that means they were here before us. They are still plentier than us, and they will continue beyond us. So that's something we need to understand, and we need to respect these microorganisms which are there, and we should learn to coexist with them, and rather than trying to destroy them. As we will be coming to in the future classes, you will realize what I'm trying to say. Now, these are the heroes which made microbiology what it is. I don't know if you've heard of any of these people. There's Robert Hooke, Virchow, Reddy, Spallanzani, Henley, Louis Pasteur, Robert Koch, Ignaz Semmelweis. So there are actually many people, but we're just trying to bring in to your focus some of the people who did such great work to bring in the concept of a microbe causing disease. And once that concept came in, then the concept of how to study it further, how to make it a scientific research, and then how to control it. So once you know how a disease is spread, and then there could be measures of stopping its spread. So when you're talking about COVID and we're saying do a social distancing, when we're saying wear a mask, wear protective equipment, what does it mean? It means that we know how it is spread, and we are trying to control it by epidemiological means, and not at this point through a vaccine, and we want to prevent it. And of course, if somebody gets infected, then we need to treat him with whatever measures we have. But that is how it originated. We got to know that there's a microbe which causes disease. We could grow it. We could understand how it spreads. And then once man advanced, we knew what drugs to give to kill it. So all these people played a big role in the advancement of medicine, and we'll be studying some of them in subsequently. What I will want you to do is to just Google these people. Since I cannot ask you questions directly, I will want you to Google these names and see what they did. And that will help you develop an interest and see and appreciate what these people did to improve the lot of life. So when we are saying that we're living in the best of stage ages, it's because of the efforts of these people who lived in the 18th century and the 19th century. And some of them, of course, in the 16th century. So these people were the game changers, and we need to address them, we need to understand them before we proceed to appreciate how they have helped us. So Anton von Leeuwenhoek described live microorganisms that he observed in teeth scrapings, rainwater, and peppercorn infusions. And he described them as little animal cues. And he is the first person who saw them and describe them. But man's knowledge was not so advanced, man's ability to correlate things were not so advanced. So it took another 200 years before one understood that these little animal cues are also associated with disease. Robert Hooke produced the first compound microscope, and he studied the plant cells in detail. And finally, we come to the big debate which happened in the 18th century, which was about origin of life. So Aristotle, a Greek philosopher, and John Needham, they were important people of the time, and they said that there is de novo generation of life, or there's spontaneous generation, that life can arise from non-living matter. And then a big fight happened because some people were thinking beyond this. They did not want to believe that life could just originate from nowhere. It's not magic that's happening. And so there was a big fight on biogenesis where Reddy and Spallanzani argued that living organisms arose from pre-existing life. It cannot be that suddenly, spoof, somebody muttered some magical words and a new life was born. They said that it has to come from a pre-existing life. Robert Virchow, again a prominent scientist of the time, said living cells arise from pre-existing cells. And then Louis Pasteur, the big father of microbiology, said that life is a germ and a germ is a life, and proved that microorganisms are present in air. So what did he do? He first put in a broth and he boiled it. So Spallanzani also did that. They boiled a broth, and then their hypothesis was that once I boiled it, I have killed whatever life there was in it, and now it should not get putrefied. There should be no fermentation. There should be no new life coming in because I've killed all old life. So Spallanzani used an open jar, and you know that open jar is open to the atmosphere, and you know that microbes are present in the atmosphere. So over time, that boiled broth got contaminated and it putrefied, and fermentation happened. So he could not exactly prove what he was saying, although he was on the right track. So what did Pasteur do? He took a conical flask, a round bottomed flask, sorry, and he boiled the broth. And after boiling it, he turned he twisted the first of all he made a long mouth of the conical flask, and not only did he make it long, but he also twisted it in such a S-shaped fashion that microbes, if they entered, would find it difficult to reach the broth. Can you understand? So it's a narrow mouth flask, and if air does enter, it's going to deposit somewhere on this twisted neck of the flask and not really reach the broth, which was sterile. So this broth stayed for months, as you can see here, and it did not get infected. It did not get contaminated, and it did not putrefy. So this was the major landmark experiment which Pasteur did, and which proved that life is not generating on its own, it needs a mother cell. Now, what was the outcome of germ theory of disease? So, this was the germ theory of disease which Louis Pasteur postulated. So, in 1840s, Ignaz Semmelweis advocated hand washing to prevent transmission of puerperal fever from one obs-gynae student patient to another. What was happening was that students were going to the morgue, were performing autopsy, and immediately after that coming to the ward and helping in the delivery of patients, and it was found that the mothers died very often. The percentage of mothers dying from birth was excessively high compared to another ward, in which the delivery was happening by midwives. So he observed the practices of these students, and he found what I just explained to you, that they came from the mortuary after autopsy. They did not wash their hands, strangely enough, and immediately proceeded to help in the delivery. So he said, why don't you wash your hands? And once that practice came in, the maternal mortality rate dropped down tremendously. And now we know that you want to prevent infections in hospitals, the one important thing we need to do is wash our hands effectively between patients. John Snow is also called the father of epidemiology, and he established that cholera was spread by fecal-oral route by a wonderful epidemiological study which you can go and read. And that's how we now know that cholera is caused by fecal-oral route and not by miasma. And Joseph Lister is the father of antisepsis. He used a chemical disinfectant to prevent surgical wound infections after looking at Pasteur's work. So people started understanding that there are microbes all over, and we need to prevent their presence to prevent infection. So surgeries were happening, but unfortunately the outcome of surgeries was very poor, because you know that microbes are all over and they were there at the site of surgery as well. And when we incise a wound, then what are we doing? We are introducing these microbes into sterile parts of the body. So what did he do? He said that before we operate, let's clean the part with an antiseptic, and that reduced the surgical site infections, reduced morbidity and mortality significantly. So Louis Pasteur is the father of microbiology, and he proposed the principles of fermentation, postulated the germ theory of disease, introduced sterilization techniques. And this is something which you must be really, really very aware of, that modern medicine, modern surgery, modern microbiology would not have advanced if we did not know the techniques of sterilization. That is, we destroy all microbes and all spores so that there can be no reinfection of, for example, microbiological media or surgical instruments and so on, so that we are able to practice asepsis. Pasteurization of milk. Now, before this technique was brought in, people were drinking unboiled milk, and that can lead to many infections like Brucella, Salmonella, Mycobacterium tuberculosis. So, the pasteurization of milk prevented infection. There's so many different types of infections. And he introduced liquid media where we could grow bacteria. Until we could grow bacteria, the science of microbiology could never have advanced. So this is liquid media, and you can see that this is clear media, and here there is bacteria growing here in this, so you can see that it is turbid as against this which is clear. He contributed to vaccine development, which is a huge development, and he developed vaccines which could prevent anthrax and rabies and fowl cholera. And you know, rabies was a disease which would lead to ultimate death. So this was a landmark discovery which Louis Pasteur did, and I'm sure you know the story about how the mother brought in this child which had been bitten by a dog, and luckily Louis Pasteur was working on rabies, and he was taking out the brains from sheep and drying them, and then trying to develop a kind of a vaccine, and that's how he saved this boy's life with the uh, vaccine which he had produced. Robert Koch is the father of bacteriology. And you know that Louis Pasteur produced liquid media, Robert Koch produced solid media, which we will be showing you. He introduced agar, which is a seaweed which helps in solidifying of microbiology media. He isolated bacteria in pure culture, and that, believe me, is the beginning of bacteriology. And this is how bacteria could look. So you can see these little colonies, this is how bacterial colonies will look. And this one colony means actually millions of bacteria living together. So one mother cell will reproduce over a period of 24 hours, and you will see these colonies of bacteria. And once I have isolated colonies, we can do so many different types of tests. We can do phenotypic tests, genotypic tests. I understand its biochemical properties, understand its antigenic properties. I'll be discussing all this subsequently with you. So with the ability to grow bacteria, he could identify tubercle bacilli, cholera bacilli, anthrax bacilli. And then he introduced the Koch's postulates, which were used to prove that a specific microbe causes a specific disease. So when once the ability to grow bacteria came in, everybody became very excited, and everybody wanted to identify the microbe or the bacteria which was causing a particular disease. So there were many people coming up with lots of theories that I know what causes diarrhea and I know what causes pneumonia. And so a set of postulates or rules had to be brought in to really confirm the hypothesis which was being laid out by so many different people. So the hypothesis or the Koch's postulates are that microorganisms are isolated from a dead animal, and then we culture it. So we will do two things. One, see under the microscope to see what is this bacteria looking like. All right, and then we will also culture onto solid medium to see what types of growth we are seeing. So it could be both ways. We first culture it, and then we see it under the microscope, but as we do routinely now, we try to see under the microscope as well as culture it. And now once the colony is grown, what do? It is grown in pure culture. We again inoculate it into an animal, and then we wait and watch to see what has happened in the animal. If the same disease is produced in the animal, and a different animal, a healthy animal, right, and the same disease, or the animal dies, and we again take out a specimen from that animal, again we culture it and see it under the microscope. If it is the same bacteria, then we say that this particular bacteria is actually causing this disease. Otherwise, the bacteria could be just present over there as an innocent bystander and not really associated with causing the disease. So, what are the Koch's postulates? The microorganism must be found in abundance in all organisms suffering from the disease. That is, it could be a human being, it could be an animal, and it should not be found in healthy organisms. The organism must be isolated from a diseased organism and grown in pure culture. The cultured microorganism should show disease when introduced into a healthy animal. And then the microorganism must be re-isolated from the inoculated, diseased experimental host and identified as being identical to the original specific causative agent. So these are the four postulates which he brought about, which played a big role in making this again into a scientific study, and where we could reject certain hypotheses and certain hypotheses were proved. And we understood which bacteria causes diphtheria, for example, or tetanus, and so on. However, as we'll be discussing in a tutorial, that this is not completely seal proof. There are always exceptions to the postulates, which we will be discussing subsequently in the tutorials. I'm sure you all know about Edward Jenner. He was the father of vaccinology, and I am sure you know the story of the milkmaid who told him that a person who is infected with the cowpox will not get infected with smallpox. And so he developed this vaccine for smallpox. And again, her mother had come and he vaccinated James Phipps with the cowpox, which causes a mild disease and makes a person immune to the more severe disease called smallpox. So now vaccines are produced from whole kill microbes, attenuated microbial strains, subunit vaccine, could be a DNA vaccine or a gene therapy. And I'm sure you've understood that you know already that there's a big race for developing a vaccine against COVID. And what is the big advantage of a vaccine is that it prevents a disease and not just cures it. It doesn't cure, it prevents the disease. So here's a cartoon which explains it, that one bug is talking to another and saying, what I really hate is preventive medicine. They go after us before we even do anything. So what is you need to know now here is that prevention is better than cure. And again, why man is so successful as a species is because of so many vaccines which prevent infection in us. So I'm sure you haven't even heard of diseases like pertussis, or you've not seen a patient with pertussis, or diphtheria, or tetanus, or polio, measles, mumps, chickenpox, because of these excellent vaccines which are there, which prevent infection altogether. Apart from vaccines which improve the lot of man, there was chemotherapy. There are antibiotics, or it could be synthetic drugs. And of course, I'm sure you know that there were always home remedies or your grandmother's remedies which could help in treating so many types of diseases. So I'm sure you know that honey can be used, ginger can be used, basil can be used. So a lot of therapies emanating from herbs have been used since centuries, and that is why humankind did not get extinct, because there were always certain remedies which would protect us from the harmful microbes. However, with the advent of synthetic or antibiotics, of course, it changed completely. Life changed completely. So the first drug was brought in 1910, which was Salvarsan, which was used to treat syphilis, which was a big problem at that time. And then sulfonamides were brought in by Gerhard Domack. A very interesting story relates to his daughter, who had got infected with a small needle, an embroidery needle. And all of us keep getting infected with stuff, we keep getting injured and so on, but nobody thinks that one would die of it. But before antibiotics were brought in, a small injury could lead to sepsis and could lead to death. So in this case, Gerhard Nomak's daughter got infected with a embroidery needle, and there would be an abscess on her arm, which the doctors would have to keep draining. But it did not work. She would keep getting fevers, she would keep getting more ill, and then they said we may have to amputate her to save her life. And even then, it was a question mark. And luckily, Tomac was working on a drug called Prontosil, and he gave it to her in desperation, and within a week she was absolutely free of the infection. And that tells you the magical status of these drugs. So antibiotics are chemicals produced by bacteria and fungi that inhibit or kill other microbes. And of course, you've all heard of the wonder drug called penicillin, which was discovered by Alexander Fleming, and it is produced by Penicillium notatum, and it inhibits a bacteria called Staphylococcus aureus. And its miraculous effect was seen firsthand in World War too, because what you will realize if you read the history of all these wars is that many people died not of the wounds that were inflicted in war, but because of the diseases or the infections they were they succumbed to because of the wounds. So millions of millions of soldiers were saved of their infections, and thus they survived. And there's again a theory that why did the Allies win against Hitler? And one of the big reasons was that they had penicillin, and they could treat their soldiers, and the soldiers could come back to combat and fight again, as against the Germans, that once you get infected, then you're bedbound and you can't really come out and fight again. So penicillin became a wonder drug of the 1940s and helped the Allies win World War II. Who are our modern day heroes? So, apart from growing bacteria and studying the phenotypic characteristics, we found that many people found the genetic basis of life. So, Watson and Crick discovered the double helix structure of DNA. Monod and Jacob discovered the operon hypothesis and the protein synthesis. We started understanding how things work on the genetic level, which I'm sure you've been introduced to in biochemistry. And then the genetic code was cracked by Khurana, Nirenberg, and Hawley. And finally, the DNA sequencing happened. And we are living in an age where we're doing whole genome sequencing, and we understand how diseases are caused, what is the genetic basis for disease, and you know that genetic engineering is happening, and we can try to prevent many genetic diseases. And not only that, we can try to understand how microbes work and how we can alter them to help us. Other modern day heroes are Carey Mullins, who discovered the polymerase chain reaction. So now we can amplify DNA, and we can look. So your COVID-19 is really being amplified by real-time PCR, which is just a modification of polymerase chain reaction, because viruses we cannot grow easily. So the best way for looking for them is by amplifying their RNA. So RNA can be converted to DNA, and which can then be amplified. And I mentioned prions earlier, which are proteinaceous infectious particles. So Prusiner discovered them. And then a very interesting search is going on, and that is the microbial basis for many diseases. And Helicobacter pylori was discovered by Marshall and Warren, and they discovered gastritis, peptic ulcer disease. And now many people are looking for a microbial reason for myocardial infarction, for multiple sclerosis, and so on. So we come to classification of bacteria, and you know that bacteria are prokaryotes. They're tiny, one-celled organisms, and actually the size is looking almost comparable, but a bacterial cell is just the size of a nucleus of a eukaryote. And you can see that it doesn't have many structures. It just has a nucleoid, which is a primitive nucleus. It doesn't have any of the organelles which are present in a eukaryote. So it doesn't have endoplasmic reticulum, mitochondria, and so on. It has a cell membrane and a cell wall. It may have a flagellum which helps it to move, and it may have a capsule. Otherwise, it just has vesicles in the cytoplasm, which help it to function in many ways. And of course, it has lots of enzymes which help it to reproduce and multiply and cause disease, that is, in the disease producing bacteria. Welcome back. I hope you had a good break. So we come back to actually serious bacteriology classification. And taxonomic hierarchy is the word we use for classifying. And why is it important that we classify? It is important because we understand the rules by which a particular group of bacteria, or any living being for that matter, works. So there's certain principles which will be common in that particular group. And then, of course, as you study them further and you go to the species level, you find that they are different in certain traits and they're similar in certain traits. So that helps us to understand a lot of things about them in a very simple way. So as far as bacteria are concerned, I'm taking the example of a gram-negative bacteria, which is called Escherichia coli. So the domain is bacteria. So all bacteria will come under the same domain, and that is bacteria. The kingdom is Monera, the same holds for all of them. Phylum is Proteobacteria. Class is Gamma Proteobacteria. Order is Enterobacteriales. Family is Enterobacteriales. The genus is Escherichia. And then finally the species is E. coli. So why I'm telling you this is that you need to know that whenever we study any living being, there is a classification, whether it's us or whether it's an animal or whether it's an insect. This is the way a classification is done. I don't want you to go into details. It's just to help you understand how the science develops. And then as per Bergey's manual, which is one of the most respected and also the oldest manual of classification, they can be classified, bacteria can be classified as basilicutes, that is, they're gram negative, they have gram negative cell walls, and they're thin skinned. Firmicutes, they have gram positive cell walls and they're thick skinned. And then there is tenoricutes, which are bacteria which lack a cell wall altogether and are soft. So you know that cell walls, you can understand from here, gives the bacteria a particular shape, gives it rigidity, and is naturally hard. Don't worry about gram negative and gram positive, I will be discussing it shortly. So coming back to classification of bacteria, it could be on the classification could be on the basis of phenotypic traits, that is what we can see, the characteristics which are visible either by microscopy, by culture, by the biochemicals and so on. And then there are genotypic traits that we go right down to the DNA, or in cases of viruses, if they are viral RNAs, we go down to the RNA and study the genetic characteristics. So what are the criteria for classifying bacteria? When you talk about the phenotypic traits, it could be microscopy, and by microscopy, what can we appreciate? We can appreciate the morphology, whether it is a rod shaped, whether it is round, whether it is spiral, or whether it is curved. We can also appreciate motility of a bacteria by microscopy. So you can see that there are rod-shaped bacteria, which is the word is used is bacilli. They could be round bacteria, where the word is cocci. They could be spiral, as you can see here, a short spiral or a long spiral, as you can observe here. It could be a comma-shaped structure. So this is the shape of an individual bacilli. Now those bacteria may be arranged in genes. So you see this kind of a picture under the microscope. They could be arranged in clusters. So you see this kind of picture. They could be arranged in pairs, or they could be bacilli arranged in long chains. So in a microscope, you can see two things. One, the shape, and second, the arrangement. That is, if I make a wet mount, and this is what I will be able to observe. So that is quite a limited information, but that's something more than nothing. So a wet mount is what you are seeing here, and this is, you can see these round structures which really belong to a fungus, and you can see that there is budding happening here and so on and so forth, but I cannot discuss much about it. Now let us look at a staining technique, and that is gram stain. And what you can see is that certain bacteria stain purple and they're called gram positive, and certain bacteria stain pink, as you can see over here. So a purple staining bacteria is called gram positive, and if you have pink staining bacteria, they call gram negative. And the stain we use here is gram stain, and we'll be discussing it in great detail in the practical lab. So on the basis of grams, you can see gram-positive cocaine clusters, you can see gram-negative cocaine pairs, and so on. So this is a very useful staining technique which helps to distinguish two different types of bacteria. And the basis of this differentiation is on the basis of the cell wall. If you remember, the gram-negative bacteria have a thin wall as against gram-positive bacteria which have a thick wall. And that is also used in the treatment of these bacteria. So if you see it under a microscope, you do get some information, but you do not get a lot of information. So the real breakthrough in microbiology came when we could actually grow the bacteria. And if you remember, Louis Pasteur was first use liquid medium, and liquid medium helps to grow the bacteria all right, and you can see that it is really turbid here in all these, and it is not turbid here, so you can understand that there is no bacteria growing here and there is bacteria growing in these tubes. But the real advantage, or the real what should I say, the real growth of microbiology really came with the advent of solid medium, when we could see them and see the cultural characteristics and isolate them and so on. So when we culture on solid medium, this is how we do it. We use a loop from a clinical specimen, and then we incubate it. So you know that once you have a specimen and you think there is a bacteria growing in it, you use a solid medium. In this case, we are using something called blood agar, and then we incubate it at 37 degrees centigrade overnight. Now the important thing you see here is that the incubation temperature is 37, because these bacteria are pathogens of man. And what is our temperature? Our temperature is 37. So these bacteria also like growing at this temperature, and if I want to grow them, I need to incubate them at 37 degrees centigrade, and I need to incubate it for 24 hours, and the next day I will be able to see bacterial colonies, as you can see here. So the big advantage of most of the bacteria is they grow very fast. So within 24 hours you'll be able to see the bacteria, as you can appreciate over here, and then I can do many tests to further identify them. So before we do that, what I would like you to see is the different types of bacterial colonies. Yeah. So this is of course what I showed you on the earlier plate, but you can see that they could be smooth, they could be rough and irregular, they could be pigmented, as you can see over here. So different types of pigments, a brown colored pigment here and a smooth mucoid bacteria here, tiny colonies, big colonies. You can see so many different types of bacteria growing on this plate. And as I explained to you earlier, a colony is one mother cell which has multiplied and cloned into so many daughter cells, which we can then see with the naked eye. And the characteristics of a colony can be described as in the shape, size, texture, pigment, opacity, convexity, emulsifiability, etc., which we will be studying later. Now, if you go it on blood agar, what you find, as I mentioned blood agar earlier, you know that blood agar will have RBCs, and what you find here is that some of the bacteria like growing on a medium containing red blood cells, and you can see that some of them utilize it or use it, and you can see this discoloration around the bacteria, clearing rather around the bacteria, and that's because they utilize the RBCs, and that means that there is hemolysis, and the word we use is hemolysis, that is, they've eaten up the RBCs, and which leads to a clearing. So you're seeing two things here. One, beta hemolysis in both. I'll be discussing beta hemolysis subsequently. Here the bacterial colony is really tiny as against the bacterial colony here. So when we distinguish different bacteria, we use different colony characteristics. One is the size, the other is the pigment, the beta hemolysis, and so on and so forth. So a pinpoint colony, as I have written over here, is this. So if you use a common pin, the pinpoint is this bacteria, very tiny with a broad zone of hemolysis, and then the other word is pinhead, and you see these large colonies with a narrow zone of hemolysis. So this is Streptococcus pyogenes, and this is Staph aureus. We'll be discussing all this. I'm just introducing you to them. Do not get worried. And then there's this interesting thing called a pigment. Some bacteria produce a pigment. So you have the green colored pigment which you're seeing over here, which is produced by a bacteria, gram-negative bacteria called Pseudomonas aeruginosa. Then you have a yellow colored pigment which is produced by Staphylococcus aureus, which is a gram-positive cocci. And then Serratia marcescens produces this beautiful red pigment, and you can see that here. So when we are classifying bacteria, we are seeing the size of the colony, we're seeing the shape of the colony, we are seeing the edges, is it entire or is it irregular, we're seeing the pigment, we're looking for beta hemolysis. Now coming to hemolysis, you can see that there are three types of hemolysis. One is alpha, in which what are we doing? We're seeing this discoloration which is greenish in color. And that is because of partial discoloration, the partial hemolysis of the RBCs. And that gives you that greenish color. And then you have beta hemolysis, which is produced by bacteria which lyse the RBC completely, and that gives you a beta hemolysis. And then there are bacteria which do not lyse the RBCs at all, that means there is no hemolysis, and these are called gamma. This type of hemolysis or no hemolysis is called gamma hemolysis. Now we come to another characteristic of bacteria, and that characteristic is whether they are fastidious or whether they are non-fastidious. What does the word fastidious mean? Fastidious means that a particular bacteria is finicky, it is very choosy, it will not grow on all types of media. So it requires a medium which has lots of nutrients. And then there is a non-fastidious bacteria which is not fussy and it can grow on very simple medium. So you have nutrient agar, which is a very basic medium, and those bacteria which grow on this medium are called non-fastidious bacteria. So Staphylococcus aureus, Pseudomonas aeruginosa, Escherichia coli, they can grow on this medium, although they cause disease, but they are non-fastidious. Then there are certain bacteria which will grow on blood agar, and they will not grow on nutrient agar. So Streptococcus pyogenes, which I mentioned earlier, then a bacteria called Streptococcus pneumoniae will grow on blood agar and not on nutrient agar. So they are more fastidious than Staphylococcus aureus. And then there are certain bacteria which require even more nutrients. So they will grow on a medium called chocolate agar and not on blood agar. Now, chocolate agar does not have chocolate, although the color is of a chocolate. It is really heated blood agar, so the RBCs are denatured and all their nutrients are released into the medium so that the bacteria can easily pick them up and grow. So the bacteria which are fastidious are those which, for example, cause meningitis. So Neisseria meningitidis, Haemophilus influenzae, Neisseria gonorrhoeae, they are highly fastidious and they will grow only on chocolate agar. Now, once the bacterial specimen has been cultured on a specific plate, I need to know at what atmosphere to incubate in. If I do not know the requirements of that bacteria, I may not be able to get any growth at all. So the incubator which I showed you earlier is used for aerobic bacteria, and what you're seeing here is the Macintosh field jar in which we put our plates for anaerobic bacteria. So what we do here is we produce anaerobiosis. We remove all the ambient air inside the jar by using a vacuum, and we pump in CO2, H2 and nitrogen combination to give it the right environment, and then only your bacteria like Clostridium tetani, Clostridium perfringens will be able to grow. Now some bacteria are microaerophilic. That is, they do not want the amount of oxygen which is present in the environment, that is 21%. They want just 5% oxygen. So then we can generate that in a very simple way, and that is by burning a candle in the jar. And when the candle gets extinguished, what has happened? More carbon dioxide has been generated and some amount of oxygen has been used, and that gives you a microaerophilic environment. Although, of course, there are many other ways of generating microaerophilic environment now, which we will be discussing subsequently with you, I think in the practical lab. Now, once I have the bacteria, okay, I need to study it further. I want to study its characteristics so that I can now differentiate the different species. So for that, one needs to be able to interpret the results and also know which test to put up. So one simple way of differentiating bacteria is growing them on differential medium. So if you remember, there was gram stain, which helped us differentiate gram-positive bacteria from gram-negative bacteria. Now we can also, then you saw a medium called blood agar, which can differentiate between beta-hemolytic colonies, alpha-hemolytic colonies, and non-hemolytic colonies. Also, you saw that chocolate agar will allow fastidious bacteria, and nutrient agar will not allow the growth of fastidious bacteria. So that is all helping in our understanding of the properties of bacteria. Now we can make the medium even more interesting and try to see if it can further differentiate bacteria. So now we have a medium which is called clad medium. It originally looks green, and when I put in a gram-negative bacteria, a non-fastidious gram-negative bacteria, and I incubate it, and the next day when I see the growth, I can see that the bacteria is either yellow, as you can see here, or it is green. Now, what does this tell me? It tells me that the bacteria is a lactose fermenter if it is yellow, okay, and it is a non-lactose fermenter if the bacteria on the plate is appearing green. Now, what is the basis of this? The basis is that some bacteria are utilizing lactose, some bacteria are not. It's their genetic makeup. How are we using this media to differentiate? We are using this media by putting in this medium a pH indicator along with lactose. So now when lactose is fermented, so the word which is used is fermentation of a sugar. Lactose is a sugar and it is fermented. Now when a sugar is fermented, what happens? The pH of the medium becomes acidic, and this indicator which we have added becomes yellow in an acidic medium. On the other hand, if lactose is not being fermented, as you are seeing over here, then the pH does not become acidic and the indicator remains the same as it was. So this is just a very basic example I've given to you on how we can differentiate different bacteria by simply growing them on intelligent media, which can help us differentiate. So we know it's a gram-negative bacteria. Now we also know it's a lactose fermenting bacteria or a non-lactose fermenting bacteria. Now, once I know it's lactose fermenting or non-lactose fermenting or a beta hemolytic or a non-beta hemolytic, I need to study further. I need to see if I can utilize its presence of enzymes or absence of enzymes to help me in understanding this bacteria. So there is an enzyme called catalase, and it is useful to differentiate between gram-positive cocci like Staphylococcus is catalase positive and Streptococcus and Enterococcus are catalase negative. So catalase is an enzyme which produces oxygen when we dip the bacteria in H2O2. So hydrogen peroxide, I dip the bacteria in it, and H2O2 breaks up into water and nascent oxygen, and then the nascent oxygen quickly combines with the other nascent oxygen, and you can see this beautiful bubbling which is happening here, which tells you that this bacteria has the enzyme catalase, as against streptococci and enterococci who do not have the enzyme catalase, so you do not see any bubbling. The same test can be done in a tube, and you can see the bubbling happening as you can see here. So Staphylococcus is catalase positive, as you can see here, and Enterococcus and Streptococci are catalase negative, which is a very useful biochemical test to differentiate between these two bacteria. Oxidase is a very useful test to differentiate gram-negative bacilli. So oxidase positive is when you see this purple color emanating when I touch the colony to the oxidase strip, and this is seen in Pseudomonas, while Escherichia coli, which belongs to the family Enterobacteriales, will give you a negative reaction. So catalase is a useful test for gram-positive cocci, and oxidase is a very useful test to differentiate gram-negative bacilli. So this I will want you to go and read, and we can discuss this in the whenever we have the time. It could be the practical, or it could be the tutorial. It's a very nice bar graph which is explaining what we have just gone through, and I'll tell you certain species which we will be discussing in future. Now, apart from growing bacteria, okay, now Some bacteria are easy to grow, some bacteria are not easy to grow. Most viruses are not easy to grow. So we can use another property, and that property is of detection of antigens. Okay, so I may not grow it, but I may be able to look for the presence of antigens, and that's equally valid for diagnosing a particular infection. So one, I could look for antigens which belong to that particular microbe, or I could look for antibodies generated in a human being as a result of infection with that particular microbe. So one test which can be done is latex agglutination, and you can see the clumping and the clearing which is happening here. One can do ELISA, which will again help us. So if the antigen is present, you will find a change in color and you will see it as yellow, and if it is not present, you will see it as blue. And they're very simple tests, and they are very useful, especially for infections where it is not easy to grow the bacteria, one, or the virus, or the bacteria will take a very long time to grow. So these can be done. Serology is a very useful tool in microbiology, and you will be taught about this subsequently. Now finally, we talk about antibiotic susceptibility. So as I told you in the beginning that we are living in the best of times. We are so technologically advanced. The life expectancy has increased from say 20 to 40, even in the early 20th century, to 70 to 80, and that is because of the introduction of antibiotics. Modern medicine has a lot of credit of modern medicine goes to the antibiotics, whether it is cancer chemotherapy, whether it is transplant medicine, whether it is any kind of immunocompromised state. We need to protect the patient from opportunistic infections, and these antibiotics help us. So how do we look for antibiotic susceptibility is in this fashion. So once I've grown the bacteria, I've identified it, I now see whether the particular bacteria is susceptible to an antibiotic or not. So these discs are antibiotic discs, and what you're seeing here is the lawn culture of the bacteria. And what you appreciate is that some antibiotics are killing the bacteria, as you can see here, that there is no bacteria growing around it. That means this particular antibiotic is working against the bacteria. As against this, there is bacteria growing right up to the antibiotic. So you know that this antibiotic is not working. So which drugs will you want to prescribe to this patient? It would be this one, or it would be this one, or it would be this one. So this is where the role of microbiology is very important. Not only are you going to tell which bacteria is causing the disease, you're also going to tell which antibiotic to give. And that brings me to a matter of concern. And that matter of concern is that although we are standing at the heights of human growth, the bacteria are fighting back, and the bacteria are fighting back spectacularly. And how are they fighting back? They're fighting back by becoming resistant to these antibiotics. And so a word called antimicrobial resistance is becoming a very, very important topic. And many people say we're reaching a post-antibiotic era. That is, we may not have many antibiotics left to cure the various infections which we were so used to ignoring. So a very important wake-up call for us. COVID has given us a wake-up call. Antimicrobial resistance is giving us a wake-up call, and we need to change the way we think and the way we are living, so that these antibiotics are preserved for future generations. Now, when we are speciating a bacteria, there are other ways of speciating it, because just biochemicals are not very useful. So we can use a tool called phage typing. And phage means a virus. So you know that viruses not only infect us, they can infect bacteria as well. And so we have one strain of bacteria over here, as you can see, and we have different viruses. In each square that you see, there's a different virus. And what you can appreciate here is that some viruses are killing the bacterial strain and some bacteria are not, and this is used as a basis of phage typing. And now we come to the cutting edge of science and technology, and that's where we are really standing now. The only limitation is cost. Otherwise, this is the future of microbiology, this is the future of medicine, and it will also bring in some amazing tools which will uh, prevent so many infections and understand why certain people are predisposed to certain diseases. What can we do to prevent certain infections and tailor medicine to the needs of that particular person? So, genotypic classification can be started by hybridization, ribotyping, amplification of DNA or RNA, sequencing, 16S rDNA sequence analysis, and microarray. This is in brief what polymerase chain reaction is. So what you can see here is the DNA target which we want to amplify. So the first thing which is done is to expose it to 94 to 96 degree temperature, and what happens at that time is that the two DNA split, and that is called denaturation. So that is the first step in polymerase, where the double helix splits. Now once it splits, the process of amplification can begin, and what you can see is that in the mix we have added the DNA primer, that is whatever we're looking for in that particular sample, we have primers for that. So at 68 degrees centigrade, now that of course depends on different amplification cycles. In this particular cycle at 68 degrees centigrade, the primer bound to the target. That is the first step in the amplification. So the primer has bound, and once it has bound, we again increase the temperature to 72 degrees centigrade, as you can see here, and that is the ideal temperature for the extension to take place. So the primer bound at 68 degrees in this particular case, and then for extension to happen, the temperature becomes around 72, and the entire DNA, original DNA, has been replicated. Now once that cycle is complete, uh, the same cycle as described earlier, cycle 1, 2, 3, is repeated. So there will be denaturation, there will be annealing, there will be extension, and you can see the multiple copies being produced. So this is one of the ideal ways by which one can amplify without the hassle of culture, without the hassle of infection. And as I already said, that COVID, the test which is being done for COVID is exactly this. Before I end, let us just see another interesting aspect of microbes, and that is the gut microbiome. So, you know that lots of bacteria are living on us, and the majority of them are living in the gut. And a new study has come up which says that if you have a good microbiome in the gut, then there's going to be reduced anxiety and stress, reduced eczema, reduced asthma, depression, diabetes. And if we continue with the kind of lifestyle we're living, it could lead to more obesity, more mental health problems, more cancer. So then lifestyle issues are related to a personal individual as well. So if we're eating healthy, we are healthier, we are happier, we have less anxiety, we develop less disease. And on the other hand, if we continue with the fast food culture, we may land up with many more problems. So to conclude, in the pre-antibiotic era, we understood the relationship between microbes and disease and also health. We understood that there are certain things which make us healthy. So our ancestors have been having yogurt and they were aware that this is something which gives us health. People understood the scientific basis of microbes and fermentation and how they're used in food and beverages. People understood immunity as well in the pre-antibiotic era and understood that we have some ways, some innate immunity which protects us. Vaccinology, as was brought in by Louis Pasteur and Edward Jenner, reinforced the concept of prevention is better than cure. Industrial revolution was also happening at the same time, and a lot of new knowledge was coming in, and standards of living had already improved health and sanitation. So, with the Industrial Revolution, there was an improvement in the prosperity of man, which led to improvement in standard of living, which led to improved health and sanitation. So, the life expectancy had already started increasing with knowledge about how disease is spread and better standard of living. Then the brave new age of the antibiotic era came in, primarily really in the 1940s. And these drugs were really magic bullets, and man felt that the end of all infectious diseases is very close by. We could use serology for detection of antigens and antibodies. Of course, we were culturing bacteria also. And unfortunately, today we're standing in a post antibiotic era where these bullets, which were called magic bullets at that time, are they paper bullets? Are they plastic bullets? Because they don't seem to be working so well. Of course, the good thing is we do have rapid molecular assays. We're able to detect many more bacteria, viruses, fungi. But the question which remains at large is, will we be able to cure these bacteria and viruses with the rapid development of multi-drug resistance? Thank you so much. If you have any queries, feel free to connect with me. Have a good day. Bye bye.